In this video, we will study about Lac Operon. But before we begin Lac Operon, let's go through the basic concepts of genetics and then we'll dive into Lac Operon. So we know that the central dogma of biology says that from DNA, RNA is formed and from RNA, proteins are formed. The process of RNA from DNA is called transcription, and when RNA forms protein, it is called translation. The DNA in prokaryotes is polycistronic. Polycistronic DNA is the one in which when DNA forms RNA, this is DNA and this is RNA, mRNA which is formed. When on translation it will form proteins, there will be many kinds of proteins that will be formed. That is why it is called polycistronic DNA in prokaryotes. Whereas in eukaryotes, the DNA is monocistronic. Now, what is monocistronic? It means DNA will form mRNA on transcription, and on translation, when it will form proteins, it will be only one type of protein that will be formed. This is monocistronic DNA. So in prokaryotes, there is polycistronic DNA, and in eukaryotes, there is monocistronic DNA. Now let's go through Lac Operon. Now we'll start with Operon. Operon is studied under regulation of gene expression. So first let's understand what is gene expression. In central dogma of biology that we studied, we said DNA forms RNA and RNA forms protein. So when DNA finally on transcription and translation forms protein, it means the genes on the DNA are expressed. So this is gene expression. Whether they are expressed or not is known as regulation of gene expression. It means if that particular protein is needed by the body, the genes will express themselves and the protein will be formed. And if that particular protein is not needed, then genes will not express and protein will not be formed because it is not needed. There is waste of energy unnecessarily if we produce a protein which is not needed by the body at that particular moment. Now, this gene expression regulation needs to be done in both prokaryotes and eukaryotes, any organism. But we will mainly focus on prokaryotes, which is regulated by operon. Now, what is operon? Operon is a set of genes in prokaryotes which are responsible for a particular function, for one particular function, having a common promoter and operator. So we have done in prokaryotes that the DNA is polycistronic, that is from one DNA, the RNA which is formed will form multiple proteins. That is polycistronic DNA. So that is why in prokaryotes it is a set of genes which is responsible for a particular function. For one function, these particular set of genes will produce protein and it is guided by a particular common promoter and operator. We will focus on Lac Operon. Now what is Lac Operon? We will use the same definition which is of operon. Only the difference is that LAC here stands for lactose. So it is a set of genes in bacteria. Lac Operon is found in bacteria E. coli, which is responsible for one particular function. What is that function? Lactose metabolism. So for lactose metabolism, whichever proteins are needed, those sets of genes will act as operon and they are controlled by a common promoter and operator. So that is Lac Operon. Now let's understand components of Lac Operon and how it functions. Okay, now first let's go through all the components of Lac Operon. So we already studied that this Lac Operon is actually a DNA segment. So this is actually DNA on this Lac Operon, and let's go through each component of Lac Operon. So first of all, this P is known as the promoter. Promoter, like when we studied transcription, we know that promoter is the region where RNA polymerase sits and recognizes the point where the transcription should be initiated. So P is the promoter region, which is actually a set of genes on DNA, or a sequence of DNA where RNA polymerase recognizes this site and it sits so that transcription is initiated. So whether this is P or this is P, both are promoters. Then I is the regulatory gene. This is a set of genes or DNA sequences which will be transcribed. So transcribed and formed messenger RNA. We will understand it more as I'll explain you the process later. Then O is the operator. This is the on and off switch of Lac Operon, whether the protein should be formed or transcription should occur or not. This is guided by the operator. These are also DNA sequences on this DNA segment which will be either blocked or it will be open depending on lactose. Okay, and then there are these three set of genes, that is Z, Y and A, gene Z, gene Y and gene A, which will form RNA, mRNA, and then they will finally form protein on translation, where they will form different enzymes. Z will form beta galactosidase, Y will form permease, and A will form trans-acetylase. So I hope you have understood all these components. I'll repeat again. P is the promoter site, which is the sequence of DNA where RNA polymerase will recognize that site and initiate transcription. I is the regulatory gene, or we can say inhibitor. These are the sets of genes which will form messenger RNA and form repressor protein. This I'll explain later. And then O is the operator, which will turn on or off depending whether these three structural genes should be expressed or not. Then there are three structural genes, Z, Y and A. Okay, gene Z or Z, whatever you say, it produces finally after transcription and after translation then the enzyme beta galactosidase. Then gene Y produces permease and gene A produces trans-acetylase. So these are the components of Lac Operon. Now this Lac Operon this concept was given first of all in 1961 by two scientists, Francois Jacob and Jacques Monod. So it is also known as Jacob and Monod concept, based on these scientists who actually recognized this Lac Operon, and they gave this concept that the genes are expressed by Lac Operon in prokaryotes. Now let's move further. As I already mentioned that this Lac Operon is found in E. coli, and Lac stands for lactose. This I've already mentioned. Now this Lac Operon will function like it will turn on or off based on the presence or the absence of lactose. Let's understand that. Now suppose lactose is absent. So we are considering right now that the lactose is absent. It means we consider that suppose there is a petri dish here, and in that there is some glucose solution. Here there is glucose, and we put all these we can say the bacteria in this petri dish. Okay, so we can mark it with this, that there are some bacteria here. Okay, now what happens is that because glucose is the main source of energy for E. coli, it is already getting all the energy needed for its regular processes. Okay, so now what happens is this promoter site, I already said that RNA polymerase recognizes this promoter site if it has to start transcription. So RNA polymerase comes and it sits here on the promoter site. As soon as it sits on the promoter site, the repressor gene I will form mRNA. So this is mRNA which is formed. This RNA will translate and it will form a repressor protein. So this is a repressor protein which is formed. Repressor protein. So I'll explain again that first of all RNA polymerase will recognize the promoter site and it will come and sit here. As it will sit here, it will initiate transcription, and this gene I, which is the regulatory gene, also called inhibitor gene, it will form messenger RNA by transcription, and by translation it will form repressor protein. Now, as soon as this repressor protein is formed, it will move and it will bind itself to the operator region of the operon. So, here this repressor protein will bind and it will attach on the operator. As it is attached on the operator, the promoter region is blocked. So, if RNA polymerase tries to come and sit here, this region is, most of it is blocked, and so the further transcription will not be initiated. So, these genes, which is Z, Y and A, these genes cannot be transcribed, that is, no transcription or translation occurs because there is no need of it. Okay, we'll understand more that why there is no need of these enzymes, but this is what happens, that first of all, when glucose is present in the medium, glucose is the main energy source for E. coli, and so it is already getting energy, enough energy, and then when RNA polymerase comes and sits on this promoter site, it initiates transcription. Gene I will express itself to form messenger RNA, and messenger RNA will form repressor protein. This protein will bind with the operator region and block the promoter region, the next promoter region, and so RNA polymerase cannot enter this promoter region, and the remaining these three structural genes, which are Z, Y and A, they cannot be expressed, and so Lac Operon will switch off. This is one condition. Now let's consider that lactose is present. Before that, let's go through the uses of these enzymes, that what these enzymes do, what is their role. So beta galactosidase, it actually converts glucose in lactose into glucose and galactose. We know that glucose is the main source of energy in bacteria, and lactose is converted into glucose, that is, their hydrolysis is done into glucose and galactose by beta galactosidase enzyme. Permease enzyme increases the permeability for lactose in E. coli. So when permease is produced, then more and more lactose can enter inside E. coli. Okay, the permeability, the pores open. And then transacetylase, the function is known, but at this level we will not study, because for Lac Operon mainly we are focusing on these two enzymes, that is beta galactosidase and permease. Now let's consider that instead of glucose water, this E. coli is present in a glass of milk. So there is a glass of milk present. This is milk, and it has these E. coli present here inside that. Now these E. coli, they are dipped in this milk which has lactose, because milk does not have sugar, it has lactose. And then what happens is that lactose has to be converted into glucose and galactose, mainly because glucose is the source of energy. So now what happens is that this beta galactosidase, it has also one more function, that it converts lactose into allolactose. So I'll come to the point later that how this beta galactosidase is already present to produce allolactose, to convert it into allolactose. But this is what happens, that first of all this lactose is converted into allolactose. Now RNA polymerase first of all identifies the promoter site and transcription is initiated. Gene I will produce messenger RNA. Messenger RNA will form a repressor protein. This, till now, this is the repressor protein. Till now it is the same as earlier, that when lactose was absent, then also a repressor protein was formed. So now repressor protein is formed. Till now the process is same. Now this allolactose which was formed, this allolactose will go and bind with this repressor protein. As it will bind with this repressor protein, then this repressor protein will undergo some structural change, some conformational change. So when there will be a change in this repressor protein, it will not be able to bind with the operator region. So what will happen? The RNA polymerase will find that this promoter site is open and it will initiate transcription because there is no repressor protein blocking it. It means Lac Operon is on. It is turned on right now. So as soon as this RNA polymerase recognizes that the operator region is not blocked, I can initiate transcription. These three structural genes will express itself and form a messenger RNA. So this messenger RNA, which is called Lac RNA, will be formed. This Lac RNA will then give rise to these three enzymes on translation. So translation will occur from Lac mRNA and all the three enzymes, that is beta galactosidase, permease and trans-acetylase, will be formed. Beta galactosidase will quickly start the hydrolysis of lactose and it will form glucose and galactose. So as soon as glucose is produced, the E. coli, all the functioning will start, and permease will increase the permeability for lactose. So more and more lactose will enter into E. coli and more hydrolysis will occur by beta galactosidase, and this process will go on. So if there is a question that if they are these bacteria, they are dipped in milk, then how long will this process go? So it will go on till it is getting the lactose. So as the lactose is being provided, this process will continue. So this is one thing. The other thing is that how is lactose converted into allolactose in the beginning when there was no beta galactosidase, because in the beginning Lac Operon was switched off. The reason being that Lac Operon is never completely switched off. It always little bit of structural gene will be expressed, that is gene Z, Y and A will, even if the Lac Operon is switched off, even if it is blocked, little bit of Z, Y and A will express and produce small quantity of these three enzymes. So these small quantity of these enzymes will help in allowing the entrance of lactose, and then hydrolysis and even allolactose is formed. So that is how it functions, that even if Lac Operon is switched off, very small quantity of these enzymes are produced so that lactose can enter, convert into allolactose, and then once it is expressed, then the process is smooth. So this is about Lac Operon. I'll again go through all the components of Lac Operon. It has promoter region. Promoter region are those sequence of DNA which helps in initiation of transcription. So that is the site where RNA polymerase recognizes and transcription starts, initiates. I is the repressor gene, or we can say regulatory gene, which produces mRNA, which finally forms repressor protein. Repressor protein is the structure or the protein which binds with the operator region to block it. So this repressor protein will actually block or turn the operator on or off. And then there are three structural genes, Z, Y and A. Z produces beta galactosidase, Y produces permease and A produces trans-acetylase. Beta galactosidase helps in the hydrolysis of lactose, producing glucose and galactose, glucose being the primary source of E. coli for energy. And then permease increases the permeability for lactose. So the permeability increases by enzyme permease, and trans-acetylase has a function, but at this level we will not study in the functioning of Lac Operon. So I hope you have understood. And one more thing, in this Lac Operon, lactose acts as inducer. So there is always a question asked, what is the inducer which guides this Lac Operon? So inducer is lactose. Lactose is the inducer. Its presence and absence affects the functioning of Lac Operon. So I hope you have understood Lac Operon, all its components, all its functioning, how it is guided by the presence or the absence of lactose. If you like my videos, then do subscribe to my channel, follow and save for more. Thanks for watching. Bye.